To provide us with the laboratory's perspective on our STD, epi, and care, we have Mark Pandori, who completed undergraduate studies in genetics at UC Berkeley and graduate school in biomedical science and HIV biology at UC San Diego. He is an associate clinical professor of laboratory medicine at UCSF and the director of San Francisco's Public Health Laboratory. Dr. Pandori's presentation will be an update on laboratory diagnostics.
I do enjoy doing this every year, and I actually genuinely appreciate it. The laboratory shares, this isn't just talk, the laboratory shares a very special relationship with STD control. Logistically, they provide most of our work for us. It's probably about 70% of the specimens that we test come from STD control or from screening or from General Hospital for chlamydia or gonorrhea. But the other reason that I appreciate our relationship so much is that STDs are really darn interesting, actually, from a scientific point of view. There's a lot of different STD organisms, but within the species there's great genetic diversity, and that provides medical challenges and scientific challenges, and for us in the lab, some diagnostic challenges as well. In short, it really makes our job quite interesting. But in terms of that diversity, I have 15 minutes, so I thought that the theme of today's topic from the lab point of view would be looking at topics within the laboratory that affect us and would affect you who work with the results, and that are based on the diversity or that variability that we see in STD organisms. I give this every year, and I started out with an hour. I think it went down to 45 minutes. Now I'm down to 15 minutes. So I'm going to have to be pretty quick about this. But I just wanted to let you know why I'm being so narrow. I could come in here and tell you all about the lab and all the new things going on in there, but I just decided to pick 3 topics that I think are really current and important to both of us. They're going to start out, I'm going to just have a few slides about an interesting situation we have with herpes simplex virus. I'm going to go over drug resistance and gonorrhea. Dr. Phillip did a marvelous job of that. I'm just going to be a little more specific about a couple of things. And then I'm going to just end talking about lymphogranuloma venereum, or LGV testing. First thing I want to talk about is herpes simplex virus and our polymerase chain reaction, which I talked actually here a couple of years ago about how we switched from culture to a molecular test for herpes simplex virus. But I want to talk about a situation we've been running into over the last year and a half, which we call the unsubtypable situation. And for those of you that use the test, I'm going to walk through this and tell you how we've conquered this problem and how we can help you understand your results better. And here's our first question. If I gave you a herpes simplex virus result that said unsubtypable, what do you think that means? That there's no herpes present in the specimen, or that there is herpes present in the specimen? And I guess it's a true-false. Correct. What that result means is that herpes simplex virus is present, but we're unable to tell you whether it's type 1 or type 2. We switched about 6 years ago now. It sounds strange to say that. From a culture-based way of looking for herpes to a DNA-based way of doing it. When we do culture, you grow the virus, actually. And then we look at, essentially, its shape to tell you whether it's 1 or 2. And we use PCR, or a DNA-based test. We're actually reading the DNA, and that DNA will tell us whether it's type 1 or type 2. The issue at hand, however, is that DNA can change over time. It can evolve, right? When we evolve, it's really DNA that's changing. And that you can also have migratory patterns of organisms with different DNA. And in the case here in San Francisco and in other locations in the United States, what we have are strains of herpes simplex virus that have come through that are unsubtypable based on the genetics that we're using to assess or to detect that virus. And so why I'm showing you this is that in the laboratory, when we get a genetic signature for 1 or 2, it will look quite clear to us. And this is exactly what our microbiologists see. But what we're getting in some instances are viruses that look perfectly in the middle. And even the geniuses in the lab can't figure this out necessarily, right? We did a retrospective study back in 2005. Actually, 15% of our herpes would fall into this category and trick our genetic test. In the Mayo Clinic, somebody actually published a study on this that where out in their jurisdiction or in their area geographically, it was more like 5%. We report these and have been reporting them as unsubtypable because we're not able to tell whether they're 1 or 2. We are possibly going to be changing that, however, to inconclusive, and I'm going to tell you why in a minute. As I told you back in 2005, about 15% of our herpes would fall into this category. In 2011, it was only 3%. So this appears to be going down. And I don't know if that's migration or evolution which is driving that change. We are, however, and I just got results this week from that section of the laboratory, we do have a new test which allows us to discern those as either 1 or 2. And in fact, we took our last 5 unsubtypables that we found at City Clinic, and we were able to show that 3 of them were herpes simplex 1 and 2 of them were 2 using this new assay. We have to do what's called a verification in order to bring that into a mode where we can report those results back to a clinician, and we are going to do that. But it looks like we've conquered this issue by using a test that looks at the genetics in a different way. In the meantime, I think that one way to resolve these is to use herpes antibody testing, right? Wasn't that right, Dr. Phillip? All right. So now we're going to switch gears. I just wanted to use this as a kind of a town hall opportunity to talk about that unsubtypable issue, but I do want to use this as an opportunity to talk about this gonorrhea issue from the lab point of view. You just heard, I think, very well in a very good presentation that we have moved from monotherapy to dual therapy for gonorrhea. And the reason that we've done that is because every pharmacological weapon that we've thrown at this organism, it has evolved a resistance against it, from penicillin to tetracyclines to fluoroquinolones, and as Dr. Phillip told you, even azithromycin when used as a monotherapy. We thought we had it licked with cephalosporins, but even that is, I'm about to tell you, a problem. We've wondered, actually, since, the late 1990s throughout the world, whether we would see resistance to cephalosporin antibiotics. And the answer is, yeah, we already have resistance to cephalosporins in the world. If we look, it was first described in Japan back in 1999 to our oral cephalosporins. Cephalosporins, as Dr. Phillip told you, are oral and injectable. By 2006, there were public health jurisdictions in Japan where 30% of their isolates were resistant to oral cephalosporins. In 2011, we had a case in Kyoto which was resistant to oral and injectable cephalosporins. And in that case and actually all of these cases, the organism wasn't just resistant to cephalosporins, these are multi-drug resistant. So that the resistance that this has, it has accumulated resistance over time. These are resistant to penicillins, tetracyclines, fluoroquinolones, and cephalosporins. The 2011 case, which was a ceftriaxone failure, was a pharyngeal case, and in that case it seemed only carbapenems and azithromycin would have worked, and it's really quite debatable, actually, given the MIC that was seen for azithromycin. Thanks for explaining MIC very well earlier. That's the minimum inhibitory concentration. It's a quantitative measure of how well a drug might work against an organism, and it's a number we acquire in the lab in vitro. The MIC to azithromycin in that case was somewhat borderline, so we don't know if azithro would have worked or not. We have seen cephalosporin resistance in other locations. In Europe, we've had failures to oral cephalosporins in Norway, England, and Wales, and also France. In Slovenia, we have had another pharyngeal failure to ceftriaxone, the injectable cephalosporin. And in Spain, we just had a paper come out which was a failure to cefixime, I believe is what it was. And in these cases, again, they were all multi-drug resistant. And what was interesting was if we genotyped the viruses that were associated with treatment failures in Europe, they were all in a family which we call ST1407. It's just a family name from a method we use to genetically type gonorrhea internationally, okay? I'm going to come back to that. Oh, I just did. Here. So if we look at City Clinic in 2011, if you looked for what we call alert values, and these would be quantitative measures of how well a drug is working, the MIC. We call it alert value, and MIC, that would be troubling. It might not necessarily mean a treatment failure, but it might mean a treatment failure could occur. We didn't have any documented treatment failures at City Clinic in 2011, but we had organisms that we found there that had alert values, which means they perhaps could have been associated with failures. 2.5% of the isolates collected at City Clinic in 2011 had ceftriaxone alert values. 0.5% were azithromycin alert values. Those were not in the same organism. They were in different organisms. So you can see why the dual treatment would be so effective, right? In terms of oral cephalosporins, a measure, cefpodoxime, which was used a few years ago in City Clinic, 7.1% of the isolates had an alert value to that drug. Interestingly, we genotyped these at the laboratory, and what we found was that their sequence type with these for these cephalosporin alert values, not the azithro, was 1407, which means that these organisms are very closely genetically related to the organisms seen in Europe and have occasionally been associated with treatment failures there. So we are in danger in the sense that the organisms with the proper genetics to have resistance are here in our city. If we pan back, we did a study in 2011 across California in collaboration with the State Department of Public Health, and we screened isolates not only from San Francisco, but also Orange County, San Diego, and Los Angeles County, which possess baseball teams that did not win the World Series this year. So when that was done, 686 isolates tested for susceptibility. We did find 34 that either had a cephalosporin or an azithromycin alert value. And a Venn diagram here illustrates how that broke down. There were 32 that were some kind of a cephalosporin alert value across California. There were 2 that were azithromycin alert values in California. And you'll note these don't meet. They do not overlap. So it means that we aren't seeing organisms that possess alert value MICs for both drugs, right, within the same creature. And that's one of the reasons why I think this dual therapy is such an important tactic in our war against this organism. So what should you do if you suspect a treatment failure? Dr. Phillip told you that you should contact STD control, and that's absolutely correct. If you want, you probably should contact us as well, because we'd like to work with you. As you heard, a culture type specimen that allows us to do culture is the best way that we can analyze the situation. That means to actually grow the organism. So that's typically done on a chocolate agar plate, which we could provide to you if you needed it. If you're at a distant location, or if you're unable to somehow work with these agar plates, there's some data that shows that we can actually collect in a swab and transport system, which is called Amy's media. And we have these at the laboratory, and I can easily have them sent out to you or mailed out, or even could ride my scooter over there and bring them to you if you need them. And that you could collect in there, and the organism will live about 24 hours in that context to allow us to culture it in the laboratory. If, however, all you have is an Aptima specimen, or let's say a NAT test specimen, we will not be able to culture it, but it doesn't mean that it's the complete end of the road. We do have genetic methods to analyze these to see if there's evidence to suggest that they may be the strains that are associated with treatment failures in Europe. The last topic here, the theme of genetic diversity in STDs, is lymphogranuloma venereum. And I want to talk about a new capability we have at the Department of Public Health Laboratory to detect lymphogranuloma venereum. Let me start with a true or false question. A specimen that is negative for chlamydia might in fact be an LGV case. True or false? Okay, so the correct answer is in fact false. LGV, lymphogranuloma venereum, is actually just a strain of chlamydia. It's what was known amongst geneticists of the organism, L. serovar. It has a tropism for lymph nodes, which means that instead of just affecting epithelial cells, it will actually get into the blood and cause a systemic infection and can get into lymph nodes by infecting what are called mononuclear phagocytes. Lymphadenitis, abscessus, buboes, proctocolitis are all symptoms that you can see. So it can cause a very serious disease. It's a very vicious form of chlamydia. It can result in a very serious systemic infection. People have, in fact, died with LGV infections. What I want to talk about is the detection of this with a genetic, at the Department of Public Health lab using PCR, and our new ability to detect, and not only detect it, but to report those results out. The way that it will work for us and for you is that if you suspect an LGV case at the clinic, it would come to our laboratory and it would be tested by our typical methods as we would test for chlamydia. If it's chlamydia positive and if you submitted the correct paperwork, which I'll just, on the requisition form, which I can help you with afterwards if you want to talk about this, the lab will process this for LGV testing, but only if it's CT positive, chlamydia positive. What we'll do is take the DNA that we used in that lab test, and then we'll run another test on it, which we got from the laboratory of Servas More in the Netherlands, and we will ask whether or not we see DNA sequence associated with LGV. So only if it is CT positive would we take that next step, and only if you suspect it. We will run the test every 2 weeks. Right now we have verified that the test only works on rectal specimens. So right now we're only accepting rectal positive CT specimens for LGV testing. We do prefer that we can run it on the Aptima vial, the same specimen that you collected for chlamydia testing, but we would prefer what is called the BD red top tube, which is also known as a universal viral transport buffer, and that's what's generally used for herpes swabs, but we can run it on either if that's all you've got. If you can't get it to the lab immediately, you can refrigerate it for 3 days. If you think you're going to have to hold on to it for more than 3 days, you should freeze it. And then, just to let you know, if the Aptima specimen that we get is negative for CT, even though you suspect LGV, we won't run it. Because as we opened with in that question, the only way that it could be LGV is if it is in fact a chlamydia. And so we don't want to use the resources to run the LGV test if we can't verify that it's chlamydia. And I think my 15 minutes are automatically up, so thank you very much.